Hello all, and welcome to what should be hopefully a really interesting lecture on DNA profiling. And we're also going to be talking about some molecular techniques that are used pretty regularly in a bunch of different labs, especially if you are in a lab that deals with DNA. All right, so we'll go ahead and get started as I share my screen. So DNA profiling. So by the end of this lecture, you should be able to, one, explain the three major steps involved that allow DNA evidence from a crime scene investigation to be used to identify suspects. You're going to learn how to visualize DNA via gel electrophoresis to predict the length of an STR or a short tandem repeat. You're going to analyze DNA gels and DNA fingerprinting graphs to match a suspect to a crime scene. And you're also going to be able to evaluate how reliable DNA profiling actually is based on the number of STRs analyzed. So how does this DNA profiling actually work? Can you actually prove someone's guilt or innocence? So before we kind of dive into like, does this work or not, I'll explain the process to you. So basically what you do is you're going to isolate DNA samples from a crime scene and from suspects. So a lot of times, you know, in crime scene TV shows, they'll like collect something at the scene. So you might collect something like blood or saliva. But sometimes they also like invite them out to have a coffee, and then they take like their coffee cup right at the end of the scene, and they're like, aha, you know, I've got some DNA. That's actually all pretty viable because you can isolate DNA from things like saliva. You're then going to make copies of specific portions of that DNA using PCR, and then you're going to analyze and compare that amplified DNA, specifically short tandem repeats from the crime scene and your suspects via either gel electrophoresis or by DNA fingerprinting. Okay, so you can think about what kind of biological specimens would yield DNA at a crime scene. So what things could you collect from someone that could actually have DNA in them? There's blood, there's saliva, semen, hair. Those specifically with hair, you need to actually make sure that you have the very end of it or the follicle. So that is what is actually like kind of attached to your head. And so when we're isolating DNA samples, we're not just looking for like the entirety of that person's genome, right? Because remember, all of your cells contain the entire genome, right? It contains the instructions for like your entire body. Your cells are just kind of specialized to use certain parts of that DNA. So just like what your cells are doing, right? They're using certain parts. We're targeting specific parts of that DNA, specifically specific genes. So you might have a target gene. And so in our previous lecture on transcription and translation, we've been talking about how, you know, when you're transcribing things, you're not transcribing the entire genome at a time, right? You might be transcribing a specific gene. And so within a specific gene, there are what is called coding and non-coding regions. So coding regions are those that actually produce an amino acid. So those are regions that get transcribed and then they get translated into an amino acid, which then if you get enough of those together, right, that creates a protein, and that protein goes to work in your body somewhere, right? Or in that cell. But there are also regions in genes called non-coding regions, and these non-coding regions are what we're going to be focusing on today. But again, all of this is within kind of your target gene, so a specific part of the DNA. Okay, so step 2 was to make copies of your specific portions of DNA using PCR. So we're not trying to make a copy of all of the DNA, we're just trying to make a copy of the portion that we're interested in or that target gene that we're interested in. So PCR stands for polymerase chain reaction. This is one of my favorite molecular techniques, mostly because it's really gratifying to do. It usually works when you do it. And also it's kind of hilariously used in crime scene TV shows. So a lot of times you're on the TV show, they'll do like a PCR in like 20 minutes. And that's kind of amazing, honestly, because PCR generally takes like 3 hours. And that's assuming like nothing goes wrong and you didn't take a ton of extra time to set up your samples. So that always kind of cracks me up. But PCR is really there to amplify specific regions and then make a whole bunch of copies for you so you can analyze them later. So if you just tried to go straight from like the crime scene to reading something out on a gel without doing PCR, it might be really difficult because you might not even have enough copies of that DNA to actually see anything. So that's really what PCR is used for in this context. One, to amplify the region you're interested in, and two, like make a crap ton of copies about copies of it, so you can actually see it on a gel. So for DNA profiling, we're really going to be focusing on these short tandem repeats, and we're going to come back to what those are. But first things first, before we move on, I'd like you guys to pause this lecture now and watch this video on PCR. Now, in addition to your videos, you also have video reflection questions in this online module. So as you're watching this video, you should be writing down the answers to those questions. All of this, of course, can be found on Sakai in your online module 2 folder. All right, step 3. You're going to analyze and compare your amplified DNA portions. So there are 2 ways that we can do this. We can kind of do it the old school way, which is known as gel electrophoresis. Now, just because it's an older technique doesn't mean it's not used. I used this particular technique in my own dissertation, and it's used all the time. And then another technique is known as DNA fingerprinting. It is just a different way to visualize kind of the same information. And it usually looks like this. Okay, so first we're going to talk about how DNA visualization by gel electrophoresis works. So first things first is you are going to make a gel. This gel is made out of agarose. It's kind of like a jelly-like substance. It kind of hardens a bit. So it kind of has this weird consistency of like Jello, but like Jello when you don't add enough water. So it's kind of tough. That's basically agarose. You load DNA at the top of the gel because DNA is negatively charged and opposites will repel. So DNA gets loaded up here. You apply an electric current, and then your DNA fragments will separate by size. And when we're talking about size of DNA, we're talking about how many base pairs or nucleotides long is that fragment of DNA. Like, is it 50 base pairs versus 100 base pairs? So if it is 50 base pairs, for example, that's a pretty short DNA fragment. So it's actually going to move much quicker through the gel than your 100 base pair fragment, which will take a longer amount of time because it's bigger. This video will detail how gel electrophoresis works. And again, you should pause the video at this point, or pause the lecture at this point to watch this video and answer questions in your video reflection assignment. Okay, so now you know how gel electrophoresis works, and we're going to talk about DNA ladders. So you're going to use DNA ladders to determine the approximate sizes of your DNA molecules. So normally, if you just put some unknown samples of DNA in, and maybe you run that gel, you may or may not know how big those fragments are without having a reference sample. So ladders, in this case, the S over here, is your reference sample. So this band is at 1400 base pairs. This one is at 600 base pairs. This one's at 300. So we're really estimating the size of the DNA molecule, and we're going to match it using the middle. So if I wanted to estimate the size of this one over here, I would be looking at the middle of this band and then dragging kind of an imaginary line across and seeing now that it's between this 300 line and this 400 base pair band, right? And I know this one is 400 because this is 600. So this one is 500 and this one is 400. So we're going like in hundreds, right? So if you counted this all the way, it'd be 1400, the next one is 1300, etc., all the way down to 600. All right, so now this question is meant for practice. So now we're going to practice reading this. So the DNA fragment indicated in lane number 2 is blank base pairs in size. So lane number 2 is over here. We're looking at this one with a little arrow. How many base pairs in size is this particular band? So if we go kind of from the middle and then we draw it all the way across, you might estimate, okay, if this one's 600, because our ladder told us that, this one must be 500, and this is kind of maybe in the middle, I don't know, like 550 base pairs. Now you can go look at your answer choices, and you'll notice that 550 doesn't exist, but 580 does, and that makes sense, right? It can't be 600 because we know it's not totally lined up. So 580 is kind of our best next guess. We can also try lane number 1. So if this is 300, we know this is 600, 400, 500, or sorry, 600, 500, 400, right? So it's kind of right in the middle between 400 and 300. So you could estimate this being about 450 base pairs. Or 350 base pairs, sorry guys. This is 400, this is 300, so this one's right in the middle at 350. All right, so that is one way to visualize your DNA fragments. Another way is to use DNA fingerprinting. Now remember, for both of these, this refers to a specific gene, like we've isolated fragments of DNA from a specific gene. So this happens to be the amylogenin gene. This is what your gel electrophoresis would look like. This is what your DNA fingerprint might look like. You'll notice that these bands line up with the peaks. So this band lines up with this peak. These 3 are also lining up with these peaks. These two, and this one, and this one. All right, so here's kind of an interactive example of how to read some DNA fingerprinting graphs. So going back to what we've learned in previous lectures, we know that males have, genetic males have an X and a Y chromosome, right? Genetic females have 2 X chromosomes. And what's interesting about this particular gene, the amylogenin gene, is that the gene on the X chromosome is actually shorter, meaning it has fewer nucleotides than the copy on the Y gene. So the Y gene, the Y amylogenin gene, is actually a little bit longer, meaning it has more nucleotides. Now, a reminder, for DNA fingerprinting, the height of these peaks don't necessarily mean something is longer or shorter. You need to be using this axis to tell you whether these nucleotides are long or short. So we can look at this first peak here. It's about 100. All right. And then you'll notice that this suspect here, sorry, that was my cat knocking her toy. Anyway, so this peak here, you'll notice is a little bit longer, so it's at like 110 nucleotides. So I just told you that the copy of the amylogenin gene on the X chromosome is actually shorter than the copy on the Y. So that means the Y has a longer copy. So this means that this is your X and this is your Y. Now you might be saying, well, why isn't there like 2 peaks, right? Because females have 2 X's. Well, remember, these X's have the same length. They're the same length. The gene is the same length on the X. So that means you're only going to have 1 peak. You're not going to have more peaks this way or this way, right? Because this is denoting nucleotide length. So in this case, A here is going to be a representative of a genetic male. However, just identifying whether a person is genetically male or female is not enough to convict someone, right? And it really shouldn't be enough to convict anyone. So to do this, so if you wanted to take this a step further and actually see if maybe their sample from the crime scene matches up with your suspect, you'd actually have to incorporate some STRs or some short tandem repeats. And that can be used as additional DNA evidence to prove maybe that they were at a particular crime scene. So this is a really important video to watch. You should be watching it, of course, and answering the questions in your video reflection notes. This video will give you an overview of how DNA fingerprinting, specifically STRs, are used in crime scene investigations and other applications. So DNA fingerprinting is not used just for crime scenes. We use it for a lot of different things, including conservation. And we will provide some more details following that video. So pause this lecture now and watch that video. All right, so hopefully you've taken a look at that video and you answered some questions so you have a good idea of how STRs actually work. Remember that STRs are DNA, stretches of DNA that are repetitive. So STRs, if you recall from the video, are found in the non-coding regions of genes. So these are regions that don't actually produce any amino acids or proteins. They are repetitive. So you can see here in this sequence, AGAT is repeated several times. The sequence of those repeated nucleotides are the same between two unrelated people. So AGAT is our repeated sequence. They would be the same between you and I. The location is also the same in the genome between two unrelated people, so also between you and I. Where you would find them in the genome, like on what chromosome, would be the same. However, what is different is the length or the number of repeats. So if you were comparing the number of repeats between two related people, so let's say you and one of your parents, that's going to be more similar than if you're comparing the number of repeats between you and me, for example, because we are unrelated. So that means someone might have 9 AGATs in a row where another person could have 17. And so that is how you tell the difference between two individuals. So this, in DNA profiling, we generally use about 14 STR regions when we're comparing suspects, and that's because the odds that two people match in all 14 regions are over 1 in a billion. So even if you're related, you're unlikely to match like your mom or your dad 100% in these regions, which makes sense. But that also means that because it's highly variable, then it's also equally unlikely to find similarities between two unrelated individuals. So it's a good way to kind of match people to a crime scene. So just a little bit of a review. So a single short tandem repeat may differ between two unrelated people by sequence length, location in the genome, or sequence and length. Think to yourself what you might put down as an answer. And in this case, the answer is length, right? So we are two unrelated people, so that's going to likely differ by length. So if we go back to this slide, you'll notice that length between individuals are highly variable, specifically if they're unrelated. Okay, so to recap, we know that comparing the STR length at the 14 different locations can detect differences in DNA identity between two people. So how would we use this information to compare people's DNA? What methods would we use? So remember, our methods include 1, isolating the DNA from the crime scene. We would then use some PCR to amplify specific STRs or specific genes. And then we would compare lengths of STRs using gel electrophoresis and DNA fingerprinting. Okay, so using STR analysis. So I've got an example here of a gel, and you can see that we've got our ladder here on the left, denoted by the L. CS refers to the crime scene, and then we have suspect A and suspect B. So we've isolated our DNA, we did our PCR, right? We amplified the specific STRs we were interested in. Now, this whole gel refers to one specific STR. So that means like if you needed more than one, you're doing all 14, you would have to run this out on 14 different gels. The STR DNA molecules are separated on this gel, so that's what we're looking at here. So now we're just going to compare the length to start. So does suspect A have more or fewer repeats at the TH01 STR site than the crime scene DNA? So here's our crime scene DNA. You can see both bands are kind of hanging out at 60 base pairs. We're going to look at suspect A and then suspect B. So we're thinking longer versus shorter. So longer length, that means you're going to have bands that cluster up here. Shorter length, you're going to have bands that cluster down here. So you'll notice that suspect A, both bands are just a little bit shifted further towards the bottom compared to suspect B. So suspect A is going to have fewer repeats than suspect B. As you can see that this is shifted slightly up, like they don't line up completely. All right, so suspect A has fewer repeats because it's a little shorter than suspect B. Especially if we're only looking at these bottom bands here, even the top bands show the same pattern, right? So this one's a little bit further down, so that means you have slightly smaller or shorter fragments than suspect B. All right, so now we are comparing across our suspects, and we have different STR markers in each of these wells. So this one happened to be looking at only one looking at specific subjects and looking at one specific marker. Now you can see that our gel is a little bit larger and you've got your markers across the top here. So that means for the crime scene, the STR markers are 1 through 6. For the suspect, 1 through 6. And then for suspect B, 1 through 6. All right, so now we're going to compare. So you'll see that if we look at marker 5 goes here, marker 4 is here for the crime scene. And this also matches up with suspect B. For number 3, it kind of looks like suspect B doesn't necessarily match completely. It does match a little, right? But you could also say suspects A's number 3 is up here. Number 1 and number 2, if we compare, you can see that, all right, that doesn't match up, but you'll see number 1 here, it matches completely, right? Number 2 is down here for the crime scene, number 2 is down here. So in this case, suspect B is matching really nicely to this crime scene. So in this scenario, with these 6 STR markers, you could probably say that suspect B was likely to have committed that crime. However, I want you guys to think about, in that analysis that I just showed you, using 6 STR markers, was that enough to convict suspect B with 100% certainty? What would you put down? Now, some of you might have said, yeah, because like, look at it, man. This matches this, right? Completely the same. However, remember I told you that there are 14 STR markers. We only looked at 6. There's a good chance that the other 6 is probably not going to match. So I don't think we can be 100% certain that suspect B was the one who committed the crime. So in this case, you would probably want to run another analysis that includes the other 7, 8. There we go. There's the math. The other 8 STR markers. Okay, so this is just another way to look at it, right? So I told you there were 2 different ways. You can use gel electrophoresis, and again, you could use DNA fingerprinting graphs. Now, this is a DNA fingerprinting graph. Now, here is our evidence sample. This is our victim reference sample. So this is the victim of the crime. This is what we pulled from the crime scene, and then this is basically kind of like a reference. Okay, so if we're looking at suspect 1 versus suspect 2, you can see that some of our stuff is lining up here, right? So these 2 peaks line up with these 2 peaks. These 3 peaks line up, these line up. These 2 don't quite line up. This one lines up, this lines up. So these are individual genes as well. So that also means that not everything is going to line up completely, because you could have different markers in different areas, right? But overall, if you compare suspect 1 and suspect 2, you'll find that suspect 2 looks really similar to our evidence sample. All right, so in this case, you might conclude that suspect 2 was more likely to have committed the crime than suspect 1. Oh, sorry, reverse that. Sorry, suspect 1 in this case, right? Because we have these two little peaks here, right, which line up really nicely. So if you compare these samples, I was looking at the wrong one. These guys, so suspect 1 is going to line up really nicely with our evidence sample compared to suspect 2. Sorry about that, guys. All right, so that is basically the end, and that's how you basically read DNA fingerprinting, and that's the end of our DNA profiling lecture. Make sure now that you finish your video reflection questions if you haven't already done so, and then move on to the next section in this module, which is on epigenetics. Other than that, I will let you guys go.